In humans, we have 46 chromosomes or we have 23 pairs of chromosomes. Each cell. So every cell that is there in our body, each of these cells will carry 46 chromosomes. Spermatogenesis is the formation of the sperms from the spermatogonia which are present in the seminiferous tubule. What is spermiogenesis? It is nothing but the process of embedding of the sperm head in the Sertoli cell. So that is nothing but spermiogenesis. Hello everyone, a warm welcome to this session on concept class for class 12th biology. I'm Dr. Divya, biology faculty. So in this session, that is session 3 of human reproduction, chapter 2, we will learn about gametogenesis. So in the previous sessions, we had learnt about the male and the female reproductive systems. In this session, we will learn about spermatogenesis and oogenesis. So talking about gametogenesis, it's a two-word, we'll split it and learn. So gametes. Gametes means the male gamete and the female gamete is what I'm talking about. So the male gamete will be the sperm. The female gamete will be the ova, or we can also call it as the egg. So here, Genesis means formation or giving rise to, birth of something. So here what is being formed? The gametes are being formed. What are the gametes that are being formed? In the male reproductive system, the male gamete, that is the sperm, is being formed. And in the female reproductive system, the female gamete, that is the ova, is being formed. So therefore, in the primary sex organ, that is the testis in the male, and in the case of female, so what are the primary sex organs, the secondary sex organs, the external genitalia, all this I have done in session 1 and session 2. So you can have a look into it. And in the females, it is the ovary. So the primary sex organ in the male is the testis. In the female, it is the ovary. So the testis produces the gametes, which is called as the sperms. And the ovary produces the gamete, which is called as the ova. Now, sperms are produced in the male and ova are produced in the female. And this process of formation of gametes, that is the sperms and the ova, we call it as gametogenesis, wherein genesis means formation. Moving further, in gametogenesis, first we'll study about the spermatogenesis. Again here, split the word, spermatogenesis. Sperma meaning production of sperms, that is genesis means production or formation. So how does the formation of sperms occur, or how sperms are produced inside the testis? We will look into. So the process of formation of sperms in males is called as spermatogenesis. So here we have the cells which are called as spermatogonia, or the sperm or the male cells, male gamete cells, or it is also called as the spermatogonia. About all that in the previous sessions we have learned. And they are diploid in nature, meaning they have complete set of chromosomes in them. They are diploid in nature and they are present where? Inside the seminiferous tubules. We had seen a lot of, that is sperm cells, right? Or male germ cells or spermatogonia inside the seminiferous tubules. And in the seminiferous tubules, they are nothing but small locule-like structures inside which a lot of, or seminiferous tubules are highly coiled structures inside which a lot of spermatogonia cells will be present. And the seminiferous tubules, that is the spermatogonia which is present in the seminiferous tubule, they will multiply by mitotic division and they will form the primary spermatocytes. Now here, they are nothing, spermatocytes are nothing but sperm cells, because cytes means cells, sperm cells, and because they undergo mitotic division, which is again equational division, the sperm cells or spermatocytes are also diploid in nature. As I told you, the spermatogonia is diploid in nature 2n. So therefore, they'll have a complete set of chromosomes. Here, the chromosome number is 46 because in humans, we have 46 chromosomes, or we have 23 pairs of chromosomes. Each cell, so every cell that is there in our body, each of these cells will carry 46 chromosomes. So therefore, the spermatogonia cell will carry how many? 46 chromosomes. That is, each spermatogonia cell that is present in the seminiferous tubule that is undergoing mitotic division will carry 46 chromosomes. Now, the spermatogonia will undergo what? It will undergo mitosis differentiation. The word differentiation, there is a difference between division means simply the cells are dividing. Okay, differentiation means they are the cells which they divide to develop into new cells. So the spermatogonia cells are dividing or undergoing equational division to develop into what? Sperm cells. Do the sperm cells look like the spermatogonia? No, they have differentiated to form the sperm cells, right? Can you see? They're different. So that is why we call it as differentiation. So they undergo mitotic differentiation and they produce the primary spermatocytes. These primary spermatocytes also are 2n in nature, meaning they also have 46 chromosomes. Why? They are undergoing mitotic division. So because they are undergoing mitotic division, same 46 set of chromosomes will be carried to the primary spermatocytes as well. Now, the primary spermatocytes then undergoes first meiotic division. Now, here it is reduction division. So, meiotic division means they'll carry only half a number of chromosomes. So, therefore, half of 46 is, meaning 46 divided by 2 is 23. So, secondary spermatocytes will have 23 set of chromosomes in each of the spermatocyte. Again, the secondary spermatocyte will undergo second meiotic division. Now, again, it is meiosis. So, therefore, they'll have again half a set of chromosome and they're called as a spermatid. So, therefore, 23. And these spermatids, finally, they will undergo differentiation to form the spermatozoa or the sperms. So, they form spermatozoa. So, this is how the process of spermatogenesis occurs. Where this happens? Inside the Sertoli cells. So, where this happens? In the seminiferous tubules. So, the cells present in the seminiferous tubules, which are nothing but spermatogonia, they undergo mitotic differentiation to form primary spermatocytes. Always remember, in the first step, they undergo mitotic division. After they form the primary spermatocytes, then they undergo meiotic division. Now, I'll tell you, always remember, this is always with 23 chromosomes, 23 pairs of chromosomes. Okay. Why it is 23? Because this is 23. It will fuse with the ova or the egg. In the egg also it will be 23. Then only it will become 46. Otherwise, what will happen? 46 plus 46 means it will be more in number, right? That shouldn't happen. So therefore, this will also have 23. The egg will also have 23. When they fuse, they'll become 46. So that is why first, though they undergo mitotic division, later on the primary spermatocytes that is formed will later on undergo meiotic division to reduce the chromosome number into half. So this is the process of spermatogenesis. So, as I told you, all these are the same explanation as I told you. So, the spermatids then transfer into the spermatozoa, which are nothing but the sperms. So, these are sperms which are haploid in number. So, what are they? They are the sperms. So, here what is being generated or what is being produced or formed? Sperms are being formed from the spermatogonia cells which are present in the seminiferous tubule. So, therefore, this process is called as spermatogenesis, formation of the sperms. Now, the sperm cell head that is there, that gets embedded in the Sertoli cells and they get released from the seminiferous tubules, and this process is called as spermiation. Now, there is a difference between spermiation and spermatogenesis. Don't get confused. Spermatogenesis is the formation of the sperms from the spermatogonia which are present in the seminiferous tubules. Okay. Spermiation is the release of the sperms. The sperms that is formed after spermatogenesis, that is released during that release of the sperms is called as the spermiation. So moving further, the spermatids that are there then gets transformed into spermatozoa, which are nothing but the sperms. And this process is called as spermiogenesis. Now, 3 words are there which you need to remember. One is spermatogenesis. The other one is spermiation. And the third one is spermiogenesis. Don't get confused with these three. What is spermatogenesis? It is the formation of the sperms inside the seminiferous tubules. That is the cells of the seminiferous tubules or the spermatogonia. That is called as spermatogenesis. What is spermiation? The release of the sperms is called as spermiation. Now, what is spermiogenesis? The spermatids which are there that gets transformed into or they get converted into sperms. That process is called as spermiogenesis. Okay. So after spermiogenesis, the sperm heads that are there, if you can see here, so these are the spermatozoa. Then we have the spermatid. The spermatid that was there, it will come and embed itself. So after spermiogenesis, the sperm heads, they get embedded in the Sertoli cells and they are finally released from the seminiferous tubules. They have to move out of the seminiferous tubules. They have to travel and they need to reach the penis, right? Because they need to get released from the seminiferous tubules because they were produced in the seminiferous tubules. That's why. And that process is called as spermiation. So now there's a difference between spermatogenesis, spermiation and spermiogenesis. Again, spermatogenesis is the formation of the sperms inside the seminiferous tubules. Spermiation is the release of the sperms from the Sertoli cells and its movement out of the seminiferous tubules. That is called as spermiation. Spermiogenesis is the transformation or conversion of the spermatids into spermatozoa, which is or conversion of the spermatids into sperms. Because once they are converted into sperms, then only they will be released from the seminiferous tubules or then only spermiation will occur. So now you got to know the difference between the three. So these processes will occur. So you can see here the same thing I showed it in in the NCERT textbook. It was given in the form of a chart or schematic representation. What did we understand? Here we have the formation of the primary spermatocyte. How did the primary spermatocyte gets formed? That is, some of the cells inside the seminiferous tubules, they got converted, or the spermatogonium that is there, they formed the primary spermatocyte. How? Through mitotic division. Mitotic division. Therefore, these are diploid, having how many? 46 chromosomes. Now they will get formed into primary spermatocyte by mitotic division. Therefore, this will also be diploid, having 46 chromosome. Now these primary spermatocyte will get converted into secondary spermatocyte by meiosis. Now if it is by meiosis, then it will be reduction division having 23 chromosomes. Next, the secondary spermatocyte will again undergo meiotic division to form the spermatids. Now this formation of spermatids will occur, which is called as spermatogenesis. Now what is this called? It's called spermatogenesis. Spermatogenesis. Next, these spermatids will get converted into spermatozoa. So spermatozoa are nothing but called as sperms. And the process is called as spermiogenesis. It's called spermiogenesis. Now I hope you understood it better. Secondary spermatocyte gets converted into spermatid. That is called as spermatogenesis. Now the spermatid gets converted into spermatozoa. It is called spermiogenesis. Now those spermatozoa or sperms get released from the seminiferous tubule. That is called as spermiation. Okay. I hope now it is clear. So we'll move further. Moving further, let's see what are the hormonal controls of spermiogenesis. So during the process of spermiogenesis, there are some hormones that are being released. So what is that? How are they controlled by the hormones is what we will look into. So spermatogenesis starts at the age of puberty due to increase in the secretion of gonadotrophin releasing hormone. So these gonadotrophin releasing hormone are secreted by the hypothalamus region of the brain. So all this you have studied in your first PUC itself. Where is the hypothalamus present? What are the hormones secreted by them and all that? So hypothalamus of the brain secretes the gonadotrophin releasing hormone, and it is secreted in males, especially when they reach the puberty. And it is at that time the formation of the sperms and all that will occur. And the increased level of gonadotrophin releasing hormone, it acts on the anterior pituitary and it promotes the anterior pituitary, or it stimulates the anterior pituitary to secrete luteinizing hormone. Apart from that, it also secretes the follicle stimulating hormone. So this luteinizing hormone that is there, it acts on the Leydig cells. So Leydig cells are also called as interstitial cells. If you remember in the previous session, we have already done this. You can have a look into it. So the luteinizing hormone, it acts on the Leydig cells and it stimulates the Leydig cells to secrete the hormone androgen. We had learned right in the previous session, the interstitial cells or the Leydig cells are secrete the male hormones, that is the androgen. Now how they secrete the androgen? To secrete the androgen, first, once the male reaches the puberty, the hypothalamus region of the brain will secrete a hormone called as gonadotrophin releasing hormone. That gonadotrophin releasing hormones, as and when it increases, it will stimulate the secretion. It will act on the anterior pituitary gland, again present in the base of the brain. It will make that gland to secrete some hormones called as luteinizing hormone and follicle stimulating hormone. The luteinizing hormone that is secreted, it will act on the Leydig cells that are present in the testes and it will stimulate those cells to secrete the hormone androgen. Now, that was a function of the luteinizing hormone. What about the follicle stimulating hormone? Follicle stimulating hormone will act on the Sertoli cells. So Sertoli cells, from that the sperms need to get released, right? It acts on the Sertoli cells and it will make the Sertoli cells secrete some factors which help in spermiogenesis. So what is spermiogenesis? It is nothing but the process of embedding of the sperm head in the Sertoli cells. So that is nothing but spermiogenesis. So for that, follicle stimulating hormone is important. So when all these processes of spermatogenesis is occurring, these hormones play a very important role. So moving further, now let's look into the structure of the sperm. We got to know that how the formation of the sperm occurs. So let's look into the structure of the sperms. So sperm, this is one very, very important question which usually appears in the exam along with the diagram. So sperm, it is made up of head, neck, middle piece and a tail. So if you can see, it has this head portion, then comes the neck region, then comes the middle piece and then comes the tail. Now in the head region, the middle piece, what all is there, we will look into. So the entire body of the sperm is covered by the plasma membrane. So this outermost layer that is there, it is nothing but the plasma membrane. So it is covered by plasma membrane. And the sperm head, if you see, it contains an elongated haploid nucleus. Why they were haploid? Because after the formation of the primary sperm, that is primary spermatocyte, they kept on undergoing meiotic division only. So that is why the nucleus is haploid. So here they'll have a haploid nucleus. So it contains a nucleus which is haploid, that containing chromosomal material. And apart from that, at the tip of the head, that is at the anterior portion of the head, you can see it is covered by a cap-like structure which is called as acrosome. And the main function of the acrosome is to direct the sperm. That is, it will tell the sperm that you have to move here so that you can go and attach to the ova or the egg in the female. So acrosome, it contains enzymes that help in fertilization of the ovum. That is, it directs the sperm or shows the path for the sperm to go and reach the ovum. And the middle piece, in the middle piece, it contains lot of mitochondria. And this mitochondria, as we know, it is nothing but the powerhouse of the cell wherein lot of ATP gets produced. How ATP gets produced, all that again, it's a previous year's lessons you have studied, right? So all that there you have studied about this. So mitochondria, a lot of energy is being produced which helps the sperm to swim. So that energy required for the tail of the sperm to move and to help it to swim is provided by the mitochondria which is present entirely in the middle piece. So that is the function. So middle piece possesses numerous mitochondria that produces energy that is needed for the movement of the tail so that the sperm can go to the ova and fix itself there properly. And the seminal plasma along with sperms constitute the semen. So this is about the sperm. So sperms, there will be millions of sperms which are present in a jelly-like fluid substance. So that jelly-like fluid substance is nothing but called as the seminal plasma. So the sperm consists of the seminal plasma also. So this is about the structure of the sperm. So next, let's look into oogenesis. So we studied about what happens inside the male testes, how the formation of the sperm or the spermatogenesis happens, all that we got to know. Now let's look into oogenesis. It's about the female. That is what happens in the female reproductive system, how the egg is produced, we will look into. Now this is oogenesis. So oogenesis is nothing but the formation of a mature female gamete. So here it is a ova. That is why it is called as oogenesis. So oogenesis is initiated during the embryo development. So spermatogenesis occurs once the male reaches the puberty, okay? But oogenesis or the formation of the egg starts in the female right from during the embryonic development itself. So oogenesis is initiated during the embryonic development stage when a couple of millions of gamete mother cells. So what are the gamete mother cells called? They are called as oogonia. What are the sperm mother cells called? Spermatogonia. And those spermatogonia got converted into what? Into sperms. Oogonia or the gamete mother cells will get converted into what? Into ova or egg. So the couple of millions of oogonia that is formed within each fetal ovary. No more oogonia are formed and added after birth. So whatever oogonia is being formed, that will be formed in the females right in the fetal stage, that is inside the mother's womb itself, when the baby is inside the mother's womb itself. Once the baby is delivered or after the baby, then no more oogonia will get formed. So this is about oogenesis. Now what happens in oogenesis we will look into. So oogenesis, here also the cells, that is the oogonia. So oogonia is nothing but the gamete mother cells. So these cells, that is the gamete mother cells that are there, they will start division and they enter into prophase 1 of meiotic division. And prophase 1 of meiotic division, and they get temporarily arrested there, or they stop and entering into further stages, and they are called as the primary oocyte. Now what happens? Oogonia again, it undergoes mitotic division. So therefore here how many chromosomes it will carry? 46 chromosomes. It will undergo mitosis differentiation to form the primary oocyte. So it will form the primary oocyte. This primary oocyte, it will not divide or not undergo enter into the second stage of division as such. First, what will happen? It will remain as it is. That is, in this primary oocyte, it will be in the prophase 1. Prophase first stage, that is meiosis 1 stage. It will be there in the prophase 1 stage. Later on, it will undergo its first meiotic division and it will form the secondary oocyte. Now it will have N, that is 23 chromosomes. Then it will get converted into the ovum. So that is what will happen. So here only one ova is being produced. Now there are many oogonia. What will happen to the other oogonia? The other oogonia that is there, they will form the polar bodies and they will leave away. They will not participate in this next further steps. They'll form the polar bodies. So here it is written as fetal life. Why fetal life is written? Because the oogonia, the formation of oogonia will take place when the child is in the fetus stage itself, or in the girl is in the fetal stage itself. Once birth or childhood is done, then oogonia formation will stop. Whatever oogonia formation had happened, that will develop into the ova or the egg. So that is why here we write it as fetal stage. Then after birth or childhood, that is up till puberty, no oogonia will get formed. So that is why. But in males, it will get formed, that is the spermatogonia gets formed only during the puberty. So all that we have studied about it. So as I told you, each primary oocyte gets surrounded by a layer of granulosa cell and it is called as the primary follicle. Now, these primary follicles, that is the primary oocyte, before entering into the secondary oocyte, it forms the primary follicles. Primary follicles are nothing but the primary oocyte. They get surrounded by a layer of granulosa cells and they are called as the primary follicles. Now, a large number of these primary follicles, which are diploid in number, having 46 chromosomes, they degenerate during the phases from birth to puberty. So therefore at puberty only 60,000 to 80,000 primary follicles are left in each ovary, and out of these 60,000 to 80,000 only few will develop into the ova or the egg, and the primary follicles again will get surrounded by more layer of granulosa cells, and they will be called as the secondary follicles. And the secondary follicles are containing only 23 chromosomes because they would have formed because of the meiotic division of the primary follicles. So the secondary follicle, soon it will get transformed into tertiary follicle. If you can see here, there is the primary follicle. The primary follicle will undergo meiosis. Primary follicle, from where did the primary follicle come? The primary oocyte got surrounded by the granulosa cells to form primary follicles. They are called as primary follicles. Now, they will form into the secondary follicle, then they will form into the tertiary follicle, and from the tertiary follicle, they mature to form the Graafian follicle. So, this Graafian follicle later gets converted into the ova and they are released. Or the ova, as I told you, they are that is the tertiary follicle. It will contain a fluid-filled cavity called antrium. So, what is this antrium? In the tertiary follicle, we'll have fluid cavity. That fluid cavity which is there, it is called as antrium. These tertiary follicle will get converted into the Graafian follicle, which is matured. Once they are matured, they will start releasing the ova or the egg. And so all these are the same as I told you at this stage, that is the primary oocyte within the tertiary follicle, it will grow in size. So when do we call it as secondary follicle, tertiary follicle? When granulosa cells around these oocytes, that is the primary oocytes, keep on adding. So the primary oocyte will develop into primary follicle. Now, inside that primary follicle, what is the primary oocyte itself? It's there. Next, it will develop into secondary follicle. So, inside the secondary follicle also, again, primary oocyte only will be there. But it's called secondary follicle because a number of layer of granulosa cells will keep surrounding that. So, at this stage, that the primary oocyte within the tertiary follicle, it grows in size and it completes its first meiotic division. After it completes its first meiotic division, it will form the secondary oocyte. Now, that is, it is an unequal division resulting in the formation of a large haploid secondary oocyte and a tiny first polar body. So, secondary oocyte again, it will contain a lot of nutrient-rich cytoplasm of the primary oocyte, and the secondary oocyte, it will form a new membrane which is called as zona pellucida surrounding it, and once the zona pellucida is surrounding it, it will become mature and it forms the Graafian follicle. Now this mature Graafian follicle that is there, it will rupture to release the secondary oocyte, which is the ovum, from the ovary, and this process is called as ovulation. So here, whatever steps we studied, it was about oogenesis. Now the release of the ova is called as ovulation. So up till here, whatever formation occurs, we call it as oogenesis. Even this also, it is oogenesis. Now this process is nothing but ovulation. So what did we learn? Primary follicle or primary oocyte gets surrounded by granulosa cells. It's called as the primary follicle. Now inside the primary follicle, the primary oocyte increases in size to form the secondary molecule. The secondary molecule forms the tertiary follicle. So the tertiary follicle, it is a cell that contains a fluid cavity which is called as atrium. Now the Graafian follicle or the secondary oocyte that is there, now that tertiary follicle that is there, it will undergo its first meiotic division to form the secondary oocyte. Understood? No? From the primary oocyte to the secondary oocyte, all these processes will occur. So to make it clear, the primary oocyte, so from where did we get the primary oocyte? So you can have a look at this again. So from where did we get the primary oocyte? From the oogonia. The oogonia that was there, it underwent mitotic differentiation to form the primary oocyte. Now, until the formation of the primary oocyte, until the primary oocyte getting converted into the secondary oocyte, various steps will occur wherein the primary oocyte will get surrounded by granulosa cells to form the primary follicle. Now those primary follicles that are there, inside that what is there? Primary oocyte is only there. So to make it even more clear, we'll write it like this. There is the primary oocyte. Primary oocyte. Now this primary oocyte that is there, it gets surrounded by granulosa cells. So we call it as the primary follicle. Now this primary follicle will get converted into secondary follicle. What happens in secondary follicle? It will get surrounded by more number of granulosa cells. Which one? Primary oocyte itself. Primary oocyte itself will get surrounded by more number of granulosa cells. Then it will form the tertiary follicle. Tertiary follicle means what? Again, the primary oocyte that is there, it will get surrounded by many more number of granulosa cells. Apart from that, it will have antrium. That is, it will have a fluid cavity. These tertiary follicles that are there, or as we can say, the secondary with these tertiary follicles that are there, they will actually get converted into the secondary oocyte. That is by meiotic division. First meiosis will occur. Then these secondary oocyte will get converted into the Graafian follicle, which is the matured one. These Graafian follicles later on, as they mature, they'll start releasing the ova or the egg, and that process is called as ovulation. So this is about the ovulation process. So this was about the session, how the formation of the sperm or spermatogenesis and how the formation of the ova, that is the oogenesis, happened. So I hope you understood the session well. So we shall meet again in the coming session wherein we'll discuss about the two important topics, menstrual cycle and fertilization and implantation. So menstrual cycle is important because ovulation occurs only during the menstrual cycle. So how, at what stages and how is what we will study under this. Now we know ovulation has occurred, sperms are also being formed. Then we'll learn how the meeting of the sperm and the egg will take place, that is fertilization, and how the sperm will embed itself in the ova or the egg, which is fertilized egg that is there, will get implanted in the uterus, we will look into. So these two topics or these three important topics we'll study in the next coming session. So we'll see you in the coming session. Thank you.